Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. So now we have determination of differential leukocyte. This practical is very much important because it helps us to number the different type of leukocytes or white blood cell present in 100 leukocyte count. Now we are going to determine or going to study the different types of white blood cells that are there. So, firstly, for the white blood cells, they are granular or agranulocytes. The granulocytes, they can be remembered with this mnemonic BEN, that is B is for basophil, E is for eosinophil, N is for neutrophils, whereas agranulocytes are monocytes and lymphocytes. And to remember their concentration, I have this mnemonic, never let monkey eat bananas. So never is for neutrophil, 62%, leukocytes 30%, monocytes 5.3, this one 2.3, and basophils 0.4. Now let us have a look at the structure of these cells. The granulocytes, which we remembered through the Ben mnemonic, first of all the basophil. See, the nucleus is bilobed, this eosinophil, bilobed. Multi-lobed nucleus in neutrophil. Lymphocytes, they can be the B lymphocytes and the T lymphocytes and monocytes. Monocytes, they are converted into macrophages and they eat large malarial parasites and certain white blood cells, etc. Lymphocytes, the B cell, produce antibodies. The T cells, they are involved in combating infections by the means of their different types. By the means of their different types, which are the the helper T lymphocytes, the suppressor T lymphocytes, and the cytotoxic ones. Now, the basophils, they are involved in allergic reactions. Eosinophils, they are involved in the formation of antigen antibody complexes. And now these neutrophils, they are involved in first line of defense, or this type of defense in which they firstly combat, they do the same function as the monocyte, but they can only do it for a small number of cells and they are short living as well. So now the method that we actually use for the differential leukocyte count is the Leishman stain method. Now, firstly, the apparatus and material we need are microscope, glass slides, lancet, cotton spirit, as we all know, 70% alcohol, and then this Leishman stain. To remember this Leishman stain, we have the mnemonic EMA. E is for eosin, the eosinophilic pencil, pink dye pencil, that we have the methylene blue, which is a basic dye. If you do not know about acidic or basic dyes, I have a video on them in the histology block 1 playlist. You can watch it over there. Acetone free and water free methyl alcohol. A for acetone free and water free absolute methyl alcohol. It actually works as a glue which fixes the blood cell to the slide. Secondly, alcohol preserves the morphology and chemical status of the cell. So the Leishman stain, which is used for differential leukocyte counting, the mnemonic for it is EMA, E for eosin, M for methylene blue, and A for acetone free and water free absolute methyl alcohol. Precautions they sometimes ask: it should be thin and smooth blood film. The slide must be clean to avoid any artifacts, and staining should be very clearly done to avoid any understaining, and lens and slide contact must be carefully guarded to avoid the breaking of oil, because we will be using the oil immersion lens having the magnification of 100x. Now we have the neutrophilia and neutropenia. Neutropenia, that is decreased neutrophil count, it can occur due to certain diseases like typhoid, parathyroid, viral infections, malaria, and bone marrow. I made this lame mnemonic for this, the PTV, BM. P is for parathyroid, this is typhoid, this is viral, this is bone marrow, and this M is malaria. Now for neutrophilia, increased amount of the neutrophils. First of all, they will increase in newborn babies, in pregnancy, in stress, in menstruation, all female related things. And pathological causes, they are certain infections, and after surgery, after hemorrhage, certain burns, etc. For eosinophilia and eosinopenia, you can use this mnemonic that's written over here. It is acute pyogenic infection, stressful condition, and steroids. For eosinophilia, we have this mnemonic PASS. And this is for parasitic infection, A is for allergic condition, S is for skin condition, pass. And this one, which is for acute pyogenic infection, the stressful conditions, and steroids. Through these mnemonics, you can easily learn all the eosinophilia, eosinopenia, etc. And this is the end of our differential leukocyte counting practical. I hope you understood all of that. This is what you need to know in your OSPE.